Have you ever looked at, say, the intricate patterns on a seashell? Or maybe the way leaves spiral around a plant stem? Or even just the stripes on a zebra? And have you wondered how such complex, really beautiful forms can emerge from something as simple as a single cell? It seems almost, well, magical, doesn't it?
It really does.
Okay.
And for centuries, it was essentially treated as magical or at least deeply mysterious by scientists. But then along came Alan Turing.
Right, the man we mostly know for defining what it means for a machine to think, for code breaking.
Exactly. But he looked at these natural patterns, not just with wonder, but with a mathematician's eye. He saw something else. Not magic exactly, but maybe a hidden, elegant equation underneath it all.
Yeah, that's what we're diving into today, isn't it? This unexpected journey into a realm far beyond what Alan Turing is usually famous for.
That's right. His groundbreaking work in computer science, I mean, it frankly overshadows so much of his other genius. We're going to explore his surprising and really profoundly influential venture into theoretical biology, specifically his seminal 1952 paper, The Chemical Basis of Morphogenesis.
And our exploration today, it's based directly on excerpts from Turing's actual paper, which was published in the very prestigious Philosophical Transactions of the Royal Society of London.
And it's crucial to stress, this isn't just some historical curiosity collecting dust on a shelf, it's a foundational text. It offers a remarkably elegant, and I'd argue still incredibly relevant, explanation for how patterns and structures develop in living organisms.
So our mission today is really to unpack Turing's extraordinary ideas, to try and understand how simple chemical interactions and diffusion, just those two things, can give rise to the astonishing complexity we see all around us in nature.
And also to explore why this deep dive into biology, coming from the father of computing, is so utterly compelling. It's quite a story.
Okay, let's unpack this. So when most people hear Alan Turing, you immediately think Bletchley Park, the Enigma machine, the dawn of AI, and modern computing.
Rightly so, of course. We celebrate him for those massive contributions.
But what's less known, and it really speaks volumes about the sheer breadth of his intellect, is that his genius just wasn't confined to those areas. He was a true polymath, wasn't he?
Absolutely. He had this deep, almost childlike curiosity about the natural world. And this paper, published just 2 years before his untimely death, it wasn't just a side project.
No, it really highlights his incredible ability to apply this incredibly rigorous, abstract mathematical thinking to fields that, at first glance seem completely unrelated to computation.
He connected abstract theory to observable biological phenomena in a way that was genuinely revolutionary for his time. It really does feel like he saw the world through this unique mathematical lens, finding patterns and rules everywhere he looked.
What's fascinating here is that Turing wasn't just like dipping his toes into biology out of idle curiosity. He was tackling a truly fundamental problem, one that biologists and philosophers had wrestled with for centuries.
Which is,
How does an organism actually get its shape?
right, the process we call morphogenesis. It comes from Greek words meaning origin of shape.
Think about it. How does a seemingly uniform sort of spherical blob of cells in a growing embryo, how does that differentiate into specific tissues, organs, and ultimately a complex patterned organism?
How do you get from that single fertilized egg, which is basically a simple symmetrical cell, to something as incredibly intricate as a human being, or a zebra with its unique stripes, or a sunflower head with those precise spiral arrangements?
He was trying to answer that core question. How do these complex patterns and structures, maybe the arrangement of tentacles on a little freshwater hydra, or the dappled markings on an animal's coat, how do they arise from what starts as a relatively homogeneous, a uniform system, like that growing embryo?
Yeah, it's often called the problem of the maker's progress. How does simplicity generate complexity? Not through some, grand preordained blueprint hanging in the sky, but through the emergent properties of its own components.
Before Turing, many theories involved things like a homunculus, a tiny preformed human in the sperm, or they assumed every cell had this incredibly detailed instruction manual dictating its exact fate and position.
Which seems a bit cumbersome, biologically speaking.
Exactly. And Turing offered this elegant, dynamic alternative.
And his radical idea to tackle this was just so elegant in its simplicity. It almost sounds too good to be true until you really dig into the math behind it.
So what was it?
He proposed that patterns are generated by the dynamic interaction of just two fundamental processes, really just two.
Okay.
First, you have chemical reactions. Substances, which Turing actually quite presciently called morphogens, These substances interact. They're created, transformed, destroyed within the developing tissue. So they're not static. They're constantly in flux.
Okay, chemicals reacting.
And
And the second.
diffusion. These morphogens naturally spread out. They move from areas where there's a lot of them, high concentration, to areas where there's less, low concentration.
Yeah.
Just like, a drop of ink spreading in water.
Right, basic physics and chemistry.
Exactly. But this is where Turing's genius really shines, seeing the profound implications of combining these two seemingly basic processes.
Absolutely. The key insight, the really crucial part, was Turing proposing that an instability in a system combining these two processes, reaction and diffusion, could spontaneously lead to the formation of stable patterns, even if the system started out completely uniform, perfectly homogeneous.
Right. Think of it like this.
Mm-hmm.
Imagine a perfectly calm pond. It's surface like glass.
Okay.
If you introduce just a tiny ripple, a small, almost imperceptible disturbance under the right conditions, that tiny ripple doesn't just fade away, which is what you might expect.
Mm-hmm.
Instead, surprisingly, the interaction of the water's properties, how it flows,
Mm-hmm.
and the way that initial disturbance spreads, it can amplify it. It can turn that tiny ripple into larger, stable, repeating wave patterns.
Ah, I see. So the system itself amplifies the initial randomness.
Precisely. That's the core idea of a reaction-diffusion system.
Mm.
Local interactions, chemicals reacting right here, diffusing over time, spreading out. They can amplify these tiny random disturbances into large-scale, predictable, and importantly, stable patterns.
It's like a spontaneous ordering emerging from apparent disorder, driven just by these simple rules. No external architect guiding each ripple or each spot on a leopard.
Exactly. No architect needed.
It's quite mind-blowing when you contrast that with the traditional view of biological development. Turing really did reimagine the developing embryo.
How so?
Well, not just as a purely biological thing following some pre-programmed genetic sequence, but fundamentally as a mechanical chemical system.
Mechanical chemicals.
So
Okay.
he was looking at the physical properties of cells, their position, their velocity, their density, their elasticity, how they push and pull on each other, right alongside the chemical properties like the concentrations of these morphogens within the developing organism.
It's a truly holistic, interdisciplinary view, integrating principles from physics and chemistry directly into the heart of biology, which wasn't necessarily common then.
Not at all. So Turing conceptualized the changes in the state of a cell, or maybe a region of tissue, based on 4 interwoven factors that would determine its future development.
Okay, what are those for?
First, there are the changes of position and velocity, governed basically by Newton's laws of motion. Cells aren't just static dots. They're constantly moving, dividing, migrating, and that movement influences everything else.
Right, the mechanics of it.
Second, he considered the stresses experienced from elasticity in motion. Think about cells pushing and pulling on each other, or the whole tissue stretching and compressing. These mechanical stresses aren't just passive forces.
No, they actively influence the chemical processes happening inside those cells,
Mm-hmm.
maybe by altering cell shape or how permeable the membrane is, that sort of thing.
Exactly. Third, and this is central to his theory, are the chemical reactions themselves, where these morphogens are created, transformed, or destroyed. That's really the engine of the patterning process.
Big chemical engine.
And fourth, crucially, is the diffusion of these chemical substances as they spread through the tissue.
And the interdependence of these 4 factors is absolutely key, isn't it?
Yeah, totally. Mechanical forces influence chemical reactions, and those chemical reactions, in turn, influence mechanical changes and cell behavior. It creates this dynamic feedback loop that drives the whole pattern formation.
It's not just a one-way street like chemical signals dictating everything. It's a constant, intricate dance between physical forces and chemical concentrations.
And this built upon existing biological ideas at the time, right? Like C.H. Waddington's concept of evocators.
Right, evocators were thought of as chemicals that induce specific developmental changes, sort of like triggers.
But if Waddington's evocators were more like external signals, how did Turing's morphogens take that concept and make it something, well, entirely more dynamic, more revolutionary? He wasn't just saying these things exist, he was suggesting something much deeper about where they come from, wasn't he?
That's precisely where Turing took this monumental leap beyond the existing ideas. While Waddington's evocators triggered a change, kind of implying a pre-existing signal or instruction was needed, Turing proposed that the morphogens themselves weren't just passively signaling. They were actively creating the very patterns they would then influence.
Ah, okay. So it's not about some external set of instructions dictating where every cell goes, like a traditional architectural blueprint.
Exactly not. This implies a profoundly powerful idea. Patterns can be self-organizing.
Self-organizing.
They don't solely rely on pre-existing external instructions or rigid genetic cues that map everything out. Instead, the local interactions of these chemicals and their intrinsic diffusion properties, that's enough to spontaneously generate complex global structures.
So the morphogens aren't just messengers, they're active participants in creating the pattern itself.
Precisely. This concept of self-organization was a major conceptual shift. It moved biology away from purely deterministic, gene-centric views of development towards a more dynamic, emergent understanding.
It suggests that much of the complexity we see in nature might arise from very simple local rules just interacting over time, rather than every single detail being explicitly hardwired from the very start.
It's the difference between, say, building a house brick by brick, following a detailed diagram, and watching a crystal spontaneously form complex facets just from a solution.
Okay, so this concept of patterns emerging from simple rules is really compelling in theory. But for someone like me looking at, say, a zebra's stripes or a sunflower spiral, the connection still feels a bit abstract. Did Turing himself point to specific biological examples, even back then, where he felt his theory was actually at play, almost like a proof of concept?
Oh, absolutely, he definitely did. Turing's theory, even in its early stages and using simplified models, provided plausible mechanisms for a whole range of natural patterns.
Such as?
Well, while the paper doesn't explicitly detail, say, the spots of a leopard versus the stripes of a tiger with specific species examples, though the underlying concept certainly applies, it does mention dappled color patterns.
Dappled, okay.
And crucially, Figure 2 in the source material provides a visual representation of exactly such a pattern, derived directly from his model. These black blobs on a white background, which clearly illustrate how the theory predicts specific visual forms, like animal markings.
And the amazing thing is, the mathematics behind it can explain both spots and stripes, right?
Exactly. The mathematical parameters of the reaction diffusion system, how fast things react, how fast they spread, can be subtly tuned. Imagine one morphogen, let's call it an activator, that triggers pigment production, and another, an inhibitor, that shuts it down.
Okay.
If the activator spreads slowly, but the inhibitor spreads quickly, you tend to get isolated spots of activation.
Like a leopard.
Right. But if they spread at more similar rates, or if the tissue itself is growing in a more elongated way, you're more likely to get stripes.
Like a tiger. Wow. So it elegantly explains how such diverse patterns could arise from the same fundamental mechanism. That's incredible.
It really is. He also specifically referenced Hydra.
The little freshwater polyp.
That's the one. A fascinating creature. He posited how its tentacles, or dactylozoids as they're called, might form through this mechanism. The paper notes that if you cut off a part of a hydra,
It regenerates. Right?
amazingly well, new tentacles can emerge, and his theory provides a powerful explanation for this remarkable regenerative ability. When you cut a hydra, the existing morphogen gradients, the chemical patterns, are disrupted.
Makes sense.
But because the system is inherently self-organizing, those morphogens don't just vanish or stay chaotic. They reestablish new gradients starting from the cut surface, leading to the precise reformation of tentacles in the correct number and position. It's like a dynamic equilibrium being restored.
Wow. And he didn't stop there, did he? He linked it to even more fundamental processes.
Yes, like gastrulation in embryonic development.
Gastrulation. That's a really crucial, dramatic stage in early development, isn't it?
Absolutely. It's where a simple hollow sphere of cells, the blastula, undergoes this massive reorganization. It transforms into a more complex, multi-layered embryo with distinct germ layers, the precursors to all the different tissues and organs.
And Turing saw a connection.
He suggested this complex reorganization, this fundamental breakdown of the initial spherical symmetry, could be explained by the emergence of morphogen gradients within that early embryo. These gradients would guide cell movements and differentiation, causing cells to fold inwards or migrate to specific locations, essentially sculpting the embryo.
So it's a leap from homogeneity to organized complexity, driven by these chemical waves.
Precisely. And it wasn't just animals. He extended the theory's applicability to the plant kingdom, too.
Oh, right, the spiral.
Exactly. Complex plant structures, specifically phlotaxis.
That's the term for the arrangement of leaves, branches, seeds, and those striking, often mathematically precise spirals.
That's it. Like you see in a sunflower head, a pine cone, or the florets of a daisy. The paper suggests that these regular, often spiral patterns, which frequently follow Fibonacci numbers and the golden ratio, incredibly can arise from the interaction of morphogens controlling growth and spacing right at the plant's growing tip, the meristem.
So as new little leaves or florets emerge.
They create a local depletion of a morphogen, maybe an inhibitor. This forces the next one to form at a specific angle and distance away, leading naturally to that characteristic spiral arrangement. His theory provided this beautiful, elegant mathematical explanation for patterns that had puzzled botanists for centuries.
Okay, so if these morphogens are the ones creating the patterns, reacting, and diffusing, what's the actual role of genes in all this? Because we often think of genes as the ultimate blueprint, right? The master architects. Does this theory sort of replace that idea or does it redefine it?
It redefines it, I think, in a very elegant way.
Yeah.
Turing proposed that the function of genes in morphogenesis is purely catalytic.
Purely catalytic. What does that mean exactly?
It's a crucial distinction and a really profound insight. It means genes don't directly, say, draw the pattern or hold a complete pixel-by-pixel blueprint for the final form, as we might intuitively imagine.
Okay.
Instead, they act more like the unseen maestros controlling the orchestra of chemical reactions. Genes control the rates of the chemical reactions that produce, destroy, or modify the morphogens.
Ah, so they're controlling the process, not dictating the final picture.
Exactly. They might specify an enzyme that breaks down a particular morphogen rapidly in one area, or maybe transport a protein that helps it diffuse across cell membranes faster, or even a receptor that allows cells to respond to its presence.
So think of genes not as the architect drawing every detail of a house.
Right, but maybe more like the quality control team for the building materials, or the engineers setting the flow rates in a complex plumbing system. They don't draw the spots on the leopard directly.
No, instead they control the enzymes and proteins that act as catalysts, speeding up or slowing down the rates at which morphogens are produced, destroyed, or diffuse.
Precisely. A specific gene might make an enzyme that rapidly breaks down morphogen Y in a certain tissue, creating a valley in its concentration. Another gene might control a protein that opens channels for morphogen X to diffuse faster. It's about setting the stage and the precise rules for the chemical dance, not dictating every single step.
That provides a really crucial and elegant link between genetics, the abstract information carried by genes, and the physical manifestation of form, without needing genes to encode every single spatial detail.
Yeah, it shifts the paradigm, from a rigid blueprint to a dynamic set of rules that allow patterns to emerge.
I can imagine that for a theory this profound, something that bridges chemistry, physics, and biology so elegantly, the underlying mathematics must be incredibly complex. I've seen bits of the paper, and it's definitely filled with linear differential equations and some pretty advanced solutions.
Oh, it is. It's mathematically dense.
So what should we, as learners trying to grasp this, take away from that mathematical backbone without getting totally lost in the specific calculations?
That's a fair question. You're absolutely right. The paper is highly mathematical. It delves deep into the solutions of linear differential equations to precisely describe the behavior of these reaction diffusion systems.
Linear differential equations. Sounds intimidating.
It can sound that way, but think of it like this. Linear here basically means that the effect is directly proportional to the cause, in a simplified sense. This helps turn complex biological interactions into more predictable relationships, allowing Turing to model how small changes in conditions could lead to predictable, not just chaotic, pattern formations. It was that mathematical rigor that gave his theory its immense power and predictive capability.
So the math was essential proof.
Absolutely. The key takeaway for you, the listener, is the conclusion that this math enables. Turing rigorously demonstrated that given certain initial conditions and specific reaction diffusion parameters, things like the rates of reaction and the rates of diffusion for these morphogens stable, distinct patterns will emerge spontaneously. The math was necessary to prove how this instability arises and how it leads to predictable repeating patterns, not just random noise.
Okay, so the math proves the emergence.
And this is where the concept of wavelength comes in, used in a mathematical sense. It describes the spatial periodicity, basically, the size and spacing of these patterns.
Like ripples in a pond again?
Exactly like ripples in a pond. A wave has a certain length between its crests. In Turing's models, the chemical wavelength describes the spatial repeating pattern of the chemical concentrations, and that directly translates to the size and spacing of the emerging biological structures.
So it could predict, say, the number of segments forming in a ring of cells.
Precisely. Or the precise distance between stripes or spots on an animal's coat. The mathematics provides the predictive power. showing that these patterns aren't arbitrary but arise predictably from those underlying parameters, the reaction rates, the diffusion rates. It's the elegance of mathematics, revealing the stunning, precise elegance of nature itself.
That makes perfect sense. And to really get a handle on how these patterns emerge from something initially uniform, Turing often used simplified model systems for his calculations, didn't he? One of the most important and often discussed is the isolated ring of cells model. Can you walk us through that thought experiment and what it revealed?
Certainly, yeah, this model of an isolated ring of cells was a really brilliant simplification. It allowed Turing to study pattern formation in a controlled, almost abstract environment, stripping away some of the complexity of a whole embryo.
So what is it?
Imagine a perfect circle of cells, just a simple biological ring, perhaps representing a slice through an early embryo or a simple organism. Initially, the morphogens, these pattern-forming chemicals, X and Y, are assumed to be perfectly evenly distributed all around this ring.
Yeah.
Everything is homogeneous, perfectly uniform, no inherent pattern yet.
Okay, a blank slate ring.
Exactly. But then, if a tiny disturbance occurs, maybe a slight random fluctuation in the concentration of morphogen X at just one point, maybe due to that inherent molecular noise we talked about, or some other tiny asymmetry.
A tiny wobble in the system.
Right. The reaction diffusion system can amplify this disturbance. Instead of just fading away back into the uniform background, this tiny fluctuation grows. It destabilizes the uniformity and leads to stable, non-uniform patterns forming around the ring.
And the wavelength concept comes in here.
Precisely. The paper discusses how the wavelength of these chemical fluctuations directly relates to the number of segments or lobes, areas of high morphogen concentration, that form around the ring. For instance, if the conditions, the reaction and diffusion rates, favor a short wavelength,
You get lots of little segments.
you might see many small segments, maybe 10 or 12 bumps around the ring. But if the conditions favor a longer wavelength, fewer, larger segments will form, maybe just 2 or 4.
So by subtly changing the parameters, the rules of the chemical game.
You can predict and generate different numbers of segments or structures around that ring, from 2 lobes to 4-lobed patterns or even more complex arrangements. It's a very clear demonstration of how simple rules, amplified by instability, can lead to diverse and predictable outcomes, all starting from a perfectly uniform state.
So this idea of breakdown of symmetry is absolutely central to the whole thing, isn't it? Starting with something perfectly uniform, perfectly symmetrical, and ending up with something patterned, asymmetrical.
Fundamentally, yes.
But Turing also introduces this fascinating, much more nuanced distinction about symmetry in his paper. P-symmetry versus F-symmetry. This sounds like it gets into some really deep territory, almost the philosophy of form and fundamental physics. What did he mean by that, and why was it so important?
It is indeed a profound detail, and one that really highlights Turing's incredible foresight and his deep philosophical engagement with the problem of biological form. He notes that, yes, the breakdown of symmetry is fundamental to morphogenesis. A uniform system, which has perfect symmetry, becomes pattern and thus exhibits asymmetry. But the nuance comes in when he distinguishes between F-symmetrical and P-symmetrical objects.
Okay, break those down for us. F-symmetrical first.
F-symmetrical objects are those that have reflectional symmetry, mirror symmetry. They look exactly the same if you reflect them in a mirror. Think of a perfect square, a perfect sphere, or roughly the external appearance of a human face if you ignore minor individual asymmetries like a mole or something.
Right, like looking in a mirror.
And crucially, the laws of physics themselves are considered F-symmetrical. A physical process looks the same in a mirror image. And significantly for development, fertilized eggs, the zygotes, are also initially considered F-symmetrical, basically spherical and symmetrical.
Okay, so that's F-symmetry. What about P-symmetry?
P-symmetrical objects, on the other hand, have an equal number of left and right-handed parts, but they are not mirror images of themselves. They possess a distinct handedness or chirality.
Handedness, like our hands, left and right.
Exactly like our hands. Or think of a screw thread, or a helix like DNA. It has a direction, a spiral that can be right-handed or left-handed, and it's not superimposable on its mirror image. You can't turn a left-handed glove into a right-handed glove just by rotating it.
And organisms often have this P-symmetry.
Profoundly so, but they're often not F-symmetrical internally. Consider your own internal organs. Your heart is predominantly on the left, your liver mostly on the right. They aren't symmetrically mirrored like your arms or legs generally appear to be externally. Your DNA itself is famously a right-handed helix.
So the big, almost philosophical question he's grappling with here is, If the fundamental laws of physics are F-symmetrical, and the zygote starts as an F-symmetrical entity, how on earth do we end up with organisms that are P-symmetrical? It seems like you can't get handedness from a perfectly symmetrical starting point unless something breaks that symmetry in a specific, non-symmetrical way. Where does that initial handedness actually come from?
Precisely. This was a critical challenge for any theory of morphogenesis, and Turing tackled it with remarkable insight. He discussed the difficulty of explaining how p-symmetrical organisms can arise from f-symmetrical zygotes and f-symmetrical laws of physics if the intermediate chemical systems, the morphogens themselves, are also strictly f-symmetrical. It seems like something has to give. Where does the handedness originate?
So what was his answer?
His groundbreaking argument suggests that while the physical laws are f-symmetrical, the chemical systems, the morphogens, can themselves exhibit p-symmetry. And this isn't necessarily due to some large pre-programmed asymmetry or a specific handed gene dictating it from the start.
No. Then how?
Instead, he proposed it arises from statistical disturbances, or what he explicitly calls Brownian movement, at the molecular level.
Brownian movement, that's the random jiggling of tiny particles, right?
Exactly. The completely random jittery motion of tiny particles like individual molecules suspended in a fluid caused by constant collisions with the surrounding fluid molecules. Imagine watching a dust mote dancing wildly and randomly in a sunbeam. That's Brownian motion on a slightly larger scale.
Okay, so molecular randomness.
Turing realized that even these infinitesimally small, seemingly random molecular jiggles, which are always present, could be the initial tiny push that breaks perfect F-symmetry in a developing embryo. This subtle, unprogrammed randomness could then be amplified by the highly sensitive reaction diffusion system.
So a random molecular bump could set the direction.
Potentially, yes. If a morphogen molecule happens to diffuse slightly more in one direction just due to these random molecular collisions, or if a reaction slightly favors one chiral form of a molecule over another due to a tiny random fluctuation in concentration, that minute, almost imperceptible difference can be amplified by the whole system.
Wow. So what does this all mean for us then?
It means that even the most subtle, seemingly insignificant molecular quirks, these tiny random disturbances inherent in the physical world, can be amplified by the reaction diffusion system. They can break the initial F-symmetry and lead to the formation of p-symmetrical structures in the resulting organism.
It's a profound realization that microscopic randomness, channeled through the right dynamic system, can shape fundamental macroscopic biological structures.
It really is. It highlights the hidden order within what looks like molecular chaos. It provides a plausible mechanism for how handedness, like the direction a snail shell spirals or the left-right arrangement of our internal organs, arises in biological forms without needing some pre-existing large-scale asymmetry to kick things off. It's a truly elegant solution to a very deep problem.
That's truly fascinating that something as subtle and almost random as Brownian motion at the molecular level could potentially influence the fundamental asymmetry of an entire organism. And to really grasp how these dynamic systems work, Turing, with his characteristic mathematician's precision, often simplified them down to their core components. He focused intensely on a specific, elegant setup. What happens when just two interacting morphogens, which he simply called X and Y, dance together within a system? What kinds of patterns and even dynamic behaviors did he uncover just by tweaking their rules?
That's right. In many of his models, Turing focused on a system involving just two interacting morphogens, arbitrarily labeled X and Y. You can think of them perhaps as an activator, X, and an inhibitor, Y, though other interactions are possible.
Okay, a simple pair.
A simple pair. And by varying what he called the rate constants, essentially how fast these morphogens react with each other, for instance, how quickly X activates more X production, or how quickly Y inhibits X and the diffusion constants, how fast they spread out through the tissue, the model predicts profoundly different outcomes. It's not just about whether a pattern forms, but about the type of pattern, or even the absence of one.
So changing the speeds changes the result. What kinds of results did he find?
Well, you could have a scenario where you just get uniform concentration. No pattern forms at all. Everything remains perfectly homogeneous, like a perfectly plain, undeveloped tissue. This tends to happen when the reaction and diffusion rates are balanced in a way that any little fluctuations are quickly damped down and smoothed out.
The stable, boring state?
Pretty much. But then, with slightly different parameters, slightly different speeds, you can get stationary waves.
Stationary waves, meaning stable, fixed patterns.
Exactly. Like the spots or stripes we discussed earlier. These patterns appear in a specific location, and then they stay put. locked in place by the dynamic equilibrium of the reaction-diffusion processes. You can imagine a chemical hotspot forming, where the activator X concentrates, surrounded by an inhibitory cold zone created by the faster diffusing inhibitor, Y.
Like islands of activity in a sea of
The
inhibition.
good way to put it. And it doesn't stop there. The model also predicts traveling waves.
Traveling so the patterns move.
Yes, patterns that move or oscillate through the tissue. This could potentially lead to dynamic changes in form over time rather than just static structures. Think of pulsating waves of chemical activity that sweep across an embryo, perhaps guiding sequential cell migration or coordinating developmental events that happen one after another.
Wow, so dynamic behavior too.
Absolutely. And the source material also describes scenarios where catastrophic instability can occur.
Catastrophic instability. Sounds dramatic.
It means that sometimes very small changes in these parameters, the reaction or diffusion rates, can lead to large, sudden, irreversible shifts in the pattern. This isn't just a minor tweak. It's almost like a sudden phase transition in physics.
Like water suddenly freezing into ice.
Exactly like that. One minute you might have a uniform field, the next, boom, a complex pattern emerges, or an existing pattern shifts dramatically to a different form. In biology, this could potentially mean that a slight genetic mutation, by altering a reaction rate just a tiny bit, could cause an organism to develop, say, a completely new number of body segments, or a different pattern of limbs entirely.
Which could be a mechanism for potentially rapid evolutionary change.
Or in some cases, it might help explain why certain developmental abnormalities arise from seemingly subtle genetic variations. It shows the system can be exquisitely sensitive near these tipping points.
It's incredible that he could predict all these different behaviors just from the mathematical equations. And what's truly remarkable, especially considering the era he was working in, the early 1950s, is that he didn't just leave it at abstract equations. He actually included numerical examples and even hand-drawn figures in his paper.
Yes, which must have been incredibly laborious.
Absolutely. This was before powerful electronic computers were readily available for this kind of work, meaning these patterns were painstakingly generated, probably by hand calculation.
That's genuinely one of the most astonishing, almost heroic aspects of the paper. Turing's inclusion of these numerical examples and those hand-drawn figures, like the famous dappled pattern shown in Figure 2, was a monumental task. These computations were performed manually, largely by hand, likely taking weeks or even months for a single complex pattern simulation.
Imagine Turing, the father of modern computing, sitting there meticulously calculating these complex patterns using probably just paper and pencil, maybe a basic mechanical calculator. He was essentially acting as a human computer.
It's almost an ironic twist of fate, isn't it? But it shows his absolute dedication to demonstrating the validity and power of his theory.
And that figure too, the one showing those black blobs on a white background.
It's a direct result of these painstaking manual calculations. It visually demonstrated how his abstract mathematical equations could indeed generate patterns that look strikingly like plausible biological patterns. It moved beyond just theoretical concepts to tangible visual outcomes.
He wasn't just saying patterns could form, he was showing them forming right there on the page.
Exactly. It was a massive leap from theoretical concept to demonstrable outcome. It provided tangible visual proof that his models could produce the kind of patterns actually seen in nature. It really solidified the plausibility of his theory and made it accessible in a way that pages of equations alone wouldn't have. He showed the potential visual consequences of the math.
That tangible visual proof must have been incredibly impactful and convincing at the time, and still is. So bringing it back to the real-world impact, how does Turing's theory, all these decades later, continue to apply to biological examples, maybe with deeper context now, and what's its broader legacy?
Oh, the implications are vast, and they continue to resonate deeply in modern science. For regeneration, as we touched on with Hydra earlier,
Right, the self-organizing recovery.
the theory provides a plausible and now widely accepted mechanism for how organisms can regenerate lost parts with the correct form and number of structures. It's not just random scar tissue growth. It's an ordered recreation of the original pattern. Why? Because the self-organizing reaction diffusion system reestablishes the necessary morphogen gradients starting from the wound site, and those gradients guide the new tissue formation.
Which has implications for understanding tissue repair and maybe even regenerative medicine today.
Absolutely. And in core developmental biology, Turing's work offered this powerful and elegant alternative to the idea, which was quite prevalent then, that every single detail of an organism's development is rigidly pre-programmed in its genes, kind of strict genetic determinism.
The blueprint model.
Right. Instead, Turing proposed that simple, local chemical rules interacting dynamically could give rise to complex global forms. This shifted the focus from needing a detailed, static genetic blueprint for everything to understanding a dynamic set of instructions that generate patterns on the fly. It makes development seem much more robust and potentially adaptable.
Less like a rigid script, more like a set of rules for improvisation, maybe.
That's a great analogy.
Mm.
And in plant forms, those spiraling patterns of leaves or florets, phyllotaxis,
Which you mentioned specifically.
yes, they are discussed explicitly in the paper as another prime example that could be explained by these principles. His theory beautifully accounts for how these highly precise, often mathematical arrangements can emerge from the continuous interaction of morphogens controlling growth right at the tips of the shoots, the meristems. Modern studies still use Turing-like patterns to model the precise angles and spacing observed in plant development. It really shows the theory's broad applicability across different kingdoms of life, from microscopic polyps to complex plants, even patterns in fungi.
It truly highlights the elegance of nature's designs, doesn't it? Showing how complexity doesn't always require complex instructions, but can emerge from the dynamic interplay of simple rules. So what's the enduring legacy of this paper? Where do we see its influence today, perhaps even beyond biology? It feels like something this fundamental should extend much further.
You're absolutely right, it does. Turing's chemical basis of morphogenesis is now widely recognized as a truly foundational paper, not just in developmental biology, but also in the relatively newer field of systems biology, which looks at biological phenomena through the lens of complex interacting systems. It was a pioneering example of truly interdisciplinary thinking, seamlessly bridging mathematics, chemistry, and biology in a way that was genuinely revolutionary for its time.
And his models, these Turing
They're
patterns.
now famously known as Turing patterns, and they are still actively studied today over 70 years later. Their applications continue to expand in remarkable ways.
Where else do we see them?
Well, in biology, as we've said, they help explain phenomena like animal coat patterns, the spots, the stripes, but also patterns in fish scales, the remarkably precise formation and spacing of hair follicles on our skin, and some even propose Turing-like mechanisms might play a role in the convoluted folding patterns on the surface of the brain.
Wow, even brain structure.
Potentially, yes. But their influence extends far beyond biology. They are studied actively in physics, chemistry, material science, even geology.
Geology. How?
For instance, similar reaction to fusion principles are thought to be at play in the formation of certain types of large-scale sand ripples or dune patterns, or the characteristic pattern seen in oscillating chemical reactions in a petri dish, like the famous Belousov-Zhabotinsky reaction, which creates these beautiful moving spirals and waves of color.
Great, I've seen pictures of that.
Even potentially in the regular spacing of certain geological formations, like mineral deposits forming under specific chemical conditions. And some researchers even use Turing pattern algorithms in computer graphics to generate realistic natural textures and patterns for movies or games.
So it really underscores the universality of these emergent patterning principles across vastly different scientific domains.
It really does. It's a testament to how profoundly simple rules can lead to astonishing complexity and order, a true hidden order emerging from what might otherwise appear to be just chaos.
So we've taken quite a deep dive today into Alan Turing's lesser-known yet incredibly profound work on the chemical basis of morphogenesis. It's been a fascinating journey, I think,
It really has.
revealing how even the most intricate, beautiful, and seemingly complex patterns in nature, from the spots on a cheetah to the spirals in a sunflower head, or even the precise number of tentacles regenerating on a tiny hydra, might arise from this elegant, dynamic dance of simple chemical reactions and diffusion.
It truly shifts our perspective on how life builds itself, doesn't it?
Mm.
Moving away from that idea of a rigid, detailed blueprint towards a more dynamic, self-organizing process where patterns simply emerge from the interactions.
Yeah, this deep dive into Turing's vision really highlights that the fundamental blueprint for an organism's form isn't necessarily some detailed instruction manual dictating every single part.
No, instead it seems to be more like a dynamic set of rules for how chemicals interact and spread, allowing complexity to spontaneously arise from relative simplicity.
So here's a final thought to leave you with. If the incredible diversity and the precise beauty of life's shapes can spontaneously emerge from these simple underlying chemical and physical laws, what does this tell us about the very nature of biological creativity and about the hidden order that might exist within seeming chaos?
It's a profound question. The next time you see a patterned shell or a striped fish or even the arrangement of leaves on a stem, you might just be looking at a living, breathing, Turing pattern, a testament to the incredible insights of a mind that saw deep connections running across all of science.